Hello everyone, thank you so much for joining us this afternoon for the next instalment of our research webinar series, which is part of our Staying Connected programme. My name is Faye and I work at Ovarian Cancer Action and I'm the Research Grants and Impact Manager. Tonight, Dr Laura Tookman and Professor Iain McNeish from Imperial College London will be giving us an overview of what the BriTROC2 study is and how this collaborative effort and research aims to find more effective and personalised treatments for women with ovarian cancer. Laura is a medical oncologist at Imperial College London and trained in medical oncology at University College London and St Bartholomew's Hospital. Laura has always had an interest in ovarian cancer and was awarded a Medical Research Council clinical training fellowship and completed her PhD investigating a novel therapy for ovarian cancer at Bart's Cancer Institute. Iain is also a medical oncologist and Professor of Oncology and Head of the Division of Surgery and Cancer at Imperial College London. He's also the Director of the Ovarian Cancer Action Research Centre, and his research focuses on developing improved therapies for ovarian cancer. And he leads the OCA-funded BriTROC Translational Research Collaborative. So thank you both for taking time out of your very busy schedules to talk to our supporters about your research today. And before I hand over to Laura, you can ask any questions via the chat box and Q&A function. They'll be able to answer your questions towards the end of the session after their presentation. I'll also include some of the questions that were sent beforehand, and we kindly ask that you do not ask any personal medical questions. So thank you so much to Laura and Iain, and I'll hand over to Laura now.
Okay, hi, good evening. So my name's Laura Tookman, and I'm as was introduced, a consultant medical oncologist working at Imperial. I'm here talking today with my colleague, Professor Iain McNeish. So thank you very much for joining us today, and it's a real pleasure to talk to you all. And of course, thanks to Ovarian Cancer Action for inviting us to talk. So I'm a clinician, and I primarily treat women with gynecological cancers. But I also work with the research team, and I'm part of the team that's set up and running BriTROC2 clinical trial. So BriTROC stands for the British Translational Research Ovarian Cancer Collaborative. And this is a network which has come together to carry out important translational research across the country to help us understand ovarian cancer and ovarian cancer biology. And this study is supported by Ovarian Cancer Action and follows on from the success of BriTROC1, the first study. So today I really wanted to introduce myself and introduce the project and try to explain why I feel this is really important. I work very closely with Iain McNeish, and together we hope to describe how this work will contribute to our understanding of ovarian cancer and ultimately how we are going to use this research to develop new treatments for ovarian cancer and improve the survival for women with ovarian cancer. So the aim of BriTROC2 is to collect cancer samples from women with ovarian cancer at diagnosis and follow these women through their treatment. We know that talking about research and asking patients for samples at the time that they're going through this treatment journey is a difficult thing to ask. And so the first thing I really wanted to do was to try and explain why we think it is so important to do this. So here are the facts about ovarian cancer and the challenges that we face. So there are over 7,000 new cases of ovarian cancer per year in the UK, leading to over 4,000 deaths. The overall 5-year survival is about 45%, and 6 out of 10 of all new diagnoses are diagnosed at a late stage. And over 70% of patients who are diagnosed at an advanced stage will relapse. We need to do better. So I see women every day with ovarian cancer. I see women who've just been given their diagnosis. I see women who I'm delighted to explain their cancer has responded to treatment. But I also sadly see women who I have to explain that their cancer has returned or their cancer hasn't responded to treatment. And as clinicians looking after women with cancer, we want to make sure that every woman gets the right treatment at the right time. We want to be able to say which treatment each woman should get and when they should get it. But in the majority of cases, we do not know why one woman may respond to treatment and their cancer never returns, whereas another with apparently identical cancer may not respond to the same treatment, or the cancer grows back quickly after treatment. So we really just want to know what is the best treatment to each individual patient that is sitting in front of us. So really, can research help us answer these questions? And we believe that this is the case. And so before we discuss what we do not yet know, what I wanted to briefly outline is what do we now know about ovarian cancer biology? Because there really have been some important and exciting new developments in the understanding of ovarian cancer over the last 10 years or so. So the first thing to say is ovarian cancer is not a single disease. It's actually 5 different types of cancer, and this is really important. And the reason why this is important is if we look at the makeup of these cancers, they are all very different. And this means that they may respond differently to different treatments. So this sort of one-size-fits-all approach to ovarian cancer really needs to be reconsidered. And it's only by knowing more about ovarian cancer biology that we're going to be able to target treatments effectively. So by far, the most common type of ovarian cancer, accounting for around 70% of cases, is high-grade serous ovarian cancer. And this is the cancer that the research is going to focus on. So the next question is, what do we know at the moment about high-grade serous ovarian cancer? So the first thing to say is it probably doesn't arise in the ovary at all. The cell of origin is probably within the fallopian tubes, and abnormal cells travel down the fallopian tube to land on the ovary. And this cartoon here demonstrates how this can also lead to cancer cells spreading throughout the abdomen. And this sort of knowledge is only possible by looking at samples from women having prophylactic surgery for ovarian cancer. And this sort of knowledge has important implications for both screening of ovarian cancer and further risk-reducing surgery. But what do we also now know about the makeup of the high-grade serous cancer cells? So the only way we're going to know more about the makeup of cancer cells is by looking at a lot of ovarian cancers. And this slide shows a summary of an important study that did just that. Several hundred ovarian cancer cells were looked at, and the changes within the cancer cells were analysed. And this graph here shows the proportion of the changes that are seen across different ovarian cancers. It gives us some really useful information, but I'm just going to focus on 2 key things. The first point is, in the centre here, we've got P53. And the reason why it's in the centre is because mutations and changes within this, this gene occur in almost all ovarian cancers. The second important thing that this graph shows is that changes in BRCA1 and BRCA2 and the proportion of cancers that have these changes. Now, BRCA1 and BRCA2 are both involved in repair of DNA. So we know that cells with changes in these proteins cannot repair their DNA accurately. But we know more than that. We actually know that this pathway of DNA repair is actually changed in around 50%. So half ovarian cancers have problems repairing their DNA. So this is all very interesting to the scientists, but why do we care about this? And can this help choose the right treatment for patients? I mean, the answer to that is a little bit yes, but we do need to know more. So firstly, the most important chemotherapy that we currently have in ovarian cancer is platinum chemotherapy, or carboplatin. And what we think is that the cells that are sensitive, the cancers that respond to carboplatin chemotherapy, are more likely to have problems repairing their DNA, whereas cancers that can easily repair their DNA damage are more likely to be resistant to DNA. But what about other treatments? So PARP inhibitor treatments is a treatment that is transforming the way that we treat cancers, ovarian cancers. And we use 3 different PARP inhibitors in ovarian cancer: olaparib, niraparib, and rucaparib. And PARP inhibitors work by damaging DNA. And cells which are able to repair their DNA are not affected by PARP inhibitors, whereas cells which are unable to repair their DNA, such as those with a BRCA mutation, cannot repair their DNA and are more likely to be responsive to PARP inhibitor treatments and more likely to respond to PARP inhibitors. So knowledge about molecular characteristics and the science of cancer cells and ovarian cancer biology is really important. It's telling us a lot, but actually, even with this knowledge of BRCA mutations and problems repairing DNA, we still cannot fully explain why one person responds to treatment and another may not. So we really need to look at the cancer cells in more detail and in more novel ways. And the best way to do that, and the only way to do that, is to look at samples from women with newly diagnosed ovarian cancer and look at changes throughout treatment. And this is why the BriTROC network was created, and it's brought together centres across the UK that specialise in ovarian cancer. And this sort of research is only made possible by the amazing women who donate their samples, and also with the help and support of Ovarian Cancer Action. I'm going to hand over to Professor McNeish to describe what these studies have been in more detail.
Thank you, Laura. So a few years ago, a group of us set up BriTROC. And as you can see, the photograph at the bottom, I looked much younger then. And researching on ovarian cancer has obviously aged me somewhat. But my colleague in Cambridge, James Brenton, and I got a group of us together to try and address big questions in ovarian cancer, because we knew that there was no way we could do it on our own. We needed to collaborate with our colleagues around the UK. And so we formed BriTROC with initially 12 major ovarian cancer centres around the UK, and Ovarian Cancer Action very kindly provided us with the money and the coordination to help us set up our first study. Laura, can I have my next slide, please? So we ran BriTROC 1, which I will tell you about in a minute. And it was a great success, but we know we need to know more. So we've set up BriTROC 2. And we're going back to some of the original centres where we started our research nearly 9 years ago. So we've got the Hammersmith in West London, where Laura and I work. We've also got University College London, also just along the Euston Road, Cambridge, Oxford, Bristol, Manchester, Glasgow and Edinburgh. Now we've picked those deliberately because they're centres where a lot of women are treated for ovarian cancer, but also where there's a lot of research already going on and where we know that samples will be collected well and that we will get excellent data and excellent samples. Because as Laura said, the only way we can understand what's going on in cancer is by analysing cancers that have come from our patients. The patients are the people we're treating, and by analysing their cancer, that's how we're going to understand more of what's going on. Next slide, please, Laura. So Laura has already told you that ovarian cancer is a very difficult disease. And what we are going to discover in BriTROC 2 is how cancers evolve and change over time, and what is happening at a molecular level during that journey. And we're going to do it by asking 250 women with newly diagnosed ovarian cancer, newly diagnosed high-grade serous ovarian cancer, if they would allow us to have a piece of their cancer to analyse in my laboratory. And Laura later on will show you exactly what we're going to ask and when we're going to get samples from patients. But it's this longitudinal, this collection over time, that's really important. So we first set the idea of collecting samples from patients about 8 or 9 years ago. And we started not by taking women who were newly diagnosed, but actually taking women whose cancer had relapsed, because we wanted to address the questions and why had the cancer come back, and was the cancer when it came back different from the cancer when it was diagnosed? And believe it or not, when we set BriTROC 1 up in 2013, nobody knew how high-grade ovarian cancer changed over time. And that's partially because when ovarian cancer grows back, it grows back within the abdomen, and getting hold of samples is actually quite difficult, or we thought it was quite difficult. So when we set up BriTROC2, one of the things we wanted to do was just ask women who had cancer that had come back, could we take a biopsy of your cancer so that we could do some analysis? And I remember going to an ethics committee, and they looked at me as if I was slightly mad and said, well, why on earth would women want to give you a bit of their sample and have a biopsy just to help your research? And I said, well, I think if I explain to people why we want to understand about their cancer and why it's important to understand why cancer comes back, I think some of them might say yes. And so they kind of shrugged their shoulders and said, okay, off you go. And so lo and behold, we asked about 630 women up and down the country whether they would allow us to have a bit of their cancer when it came back. And some of them said no, or some of them said, look, I don't really feel well enough, I need to start my chemotherapy now. But 276 of them said yes. And so we got tumour samples from 276 women with recurrent ovarian cancer, purely so that we could do our research. And this was the first study actually anywhere in the world that had collected this number of samples from women with recurrent ovarian cancer. And that on its own was a fantastic achievement, because we've now demonstrated that this could be done. Next slide, please, Laura. So this was BriTROC2. Next slide, please. And I'm going to try and explain what we did. And so I'm going to move from this slide from left to right, and I'm going to try and explain about molecular biology. So I'm going to plant a rather unpleasant thought in your brain, which is your parents having sex, because that's how you came into the world. And at the moment you were conceived, you got half of your genetic material from your mother and half of your genetic material from your father. And every cell in your body, however old you are now, contains half of your genetic material from your mother and half the genetic material from your father. And so when we look at DNA in normal cells, we see two copies of everything, one maternal, one paternal, and it's exactly the same in every cell in your body. So when we do what's known as copy number, how many copies of the genes do we see from the mother and the father? The answer is always a very boring flat line where the answer is one from each. And unfortunately, we measure things in logarithmic scale, so you just have to take my word for it. When I mean naught, that actually means 2. So we have, if you take a normal cell in your body and look at the DNA and say, how much comes from your mother, how much comes from your father, and how many copies are them, you get a very flat, boring line. In the middle, you see what the DNA of one high-grade ovarian cancer looks like. And it is completely chaotic. So as we go along the bottom here, these are the chromosomes. So you have 46 chromosomes. So numbers 1 to 22, and then you have times 2, so that's 44, and then 2 sex chromosomes that determine your gender. And as you can see, in this ovarian cancer, there's bits of the DNA where the chromosomes have literally completely disappeared. So in this cancer, chromosome 13 has basically disappeared. Another bit where you've got 2, 3, 4, 5, 6, 7, 10 copies of that chromosome. So the chromosome has doubled and doubled again and doubled again. So when you look at the DNA of one ovarian cancer, it appears to be utterly chaotic. And the thing you see ticking away on the right-hand side of this graph are 118 consecutive samples we got from BriTROC cancers. And you can see 2 things from this. The first is that they are all completely crazy and chaotic. And secondly, that no 2 look the same. So we started analysing these cancers and to try and understand A, what the chaos was, and B, what did it mean. So next slide, please. So we set ourselves in BriTROC2 two very important questions. First one was, can we try and identify patterns, meaning in this chaos? And next slide, can we explain what happens when cancer comes back? So what we see here is one patient I treated many years ago. She was a 63-year-old retired school teacher, and she, like many patients, presented with advanced ovarian cancer. And many of you will know we measure a blood test called CA125, a tumour marker. And what we see here are the tumour markers of my patient over time. We treated her with carboplatin and Taxol chemotherapy, and she responded really well, and we operated. But unfortunately, about a year later, her cancer came back. And we treated her with chemotherapy, this time with carboplatin and Calyx, and she responded beautifully. But unfortunately, about a year later, her cancer came back. So we gave her some carboplatin and she responded a little bit, and then her cancer came back, and then we gave her weekly Taxol. So the next slide, go back. So the question we wanted to ask is, what is happening when the cancer responds and relapses and responds and relapses, and can we understand why it comes back? So those were the two questions we set ourselves in BriTROC2. So next slide, please, Laura. So here are the 276 women who were recruited into BriTROC. So they were recruited at the time of this vertical black line here. And as you can see, some of them were diagnosed up to 20 years before they were recruited, but most people were diagnosed around 2 years, 2.5, 3 years before we recruited them into BriTROC. And then they were treated with their chemotherapy, and we followed them for up to 4 years. Next slide, please. So how did we address the chaos question? Next slide, please. So what we did was to break down something on the left that looked apparently chaotic. And we said, okay, can we break it down into component parts that we can measure and that we think we understand? And so here's a pattern on the left that appears to be chaotic, but actually it's made up of 4 separate types of mini chaos. But when you put them together, they appear to be utterly chaotic. But we're actually able to extract from them patterns within the mess. Next slide, please. So we actually, and I won't bore you with all the details, but we actually looked for 6 different types of abnormalities, 6 little mini patterns of chaos, and then broke them down and said, okay, let's take every cancer and break it down into those 6 separate bits and see if we can measure what is going on. Next slide, please. And by analysing all of the BriTROC samples and then mining the archives for other samples, we were able to identify what we call signatures, patterns in the chaos that enabled us to unpick what was going on in our high-grade serous ovarian cancers. And each of these vertical bars is one patient, and each color is a particular type of abnormality, a particular type of mini chaos. And we discovered that each particular type of mini chaos was driven by particular mutations in the DNA that was making the cancer grow. Next slide. We discovered that the patterns were also what we call prognostic. So if your cancer had lots of green, unfortunately you did rather badly, but if your cancer had lots of purple, actually you did much better. And this is the way we're beginning to understand why some women, even though their cancers look identical, some women do much better and some women do much worse by understanding the patterns of chaos at the time of diagnosis. Next slide. The tricky bit is what happens at relapse. So here we have 120 patients at diagnosis on the left, and the same 120 patients at relapse from the BriTROC sample. And there are some subtle differences beginning to emerge, that we can see that the chaos is evolving and changing in response to treatment. It's quite hard to measure, but actually there's quite a lot more of this purple pattern here at relapse. And this is as more damage gets done to the DNA, it evolves in response to it. And that's enabling us to understand why the cancers come back and the molecular mechanisms that are driving it. Next slide, please. So here we can see in our relapse samples, if you take them all together and average them, you can find that when the cancer comes back, there's more a purple number 3 and less number 4. And that's telling us something about the processes driving the cancer forward to recur. Next slide, please. So BriTROC1 was great. It enabled us to start understanding the patterns that are going on in the cancer as they relapse. But we've actually used up all the samples from BriTROC1. It's hard to imagine that we would, but when we recruited patients into BriTROC1, we took biopsies, which are very, very small, and eventually you use all the sample up. So we've, as it were, run out of DNA. But also, we've realized there was an intrinsic problem, in as much as by recruiting women when they'd relapsed, we were recruiting women, weirdly, who actually had relatively good prognosis. And the only way to look at this properly is to take a completely unselected group of women at the time of diagnosis and follow them forwards. That also enables us to get more DNA, because we can get samples when women have surgery. And it also enables us to do a wider range of molecular analyses, because we can get more DNA from the samples. And as I said, we can follow them forwards. So BriTROC2 will enable us to start answering more questions and answer those questions in more detail, but more, it will give us that picture of evolution over time in a whole cohort of women are going through the treatment at the same time. Next slide, please. So this is what we're going to ask patients to do, and we'll be interested in your thoughts. So every woman who is diagnosed with ovarian cancer has to have the diagnosis confirmed, because we can't treat them unless we know that they've definitely got cancer. So as you will know, many women are referred to us with the suspicion that they've got ovarian cancer. And so they have a biopsy that the pathologist looks at to confirm that diagnosis. And we're going to ask women, well, if you're having a biopsy, could we just take a little bit of it to do our research with? Then the women often will have chemotherapy before their surgery. So we're going to ask them for some blood samples, because in the blood of women with ovarian cancer, we can find little bits of DNA that have, as it were, leached out of the cancer. So we can start analyzing the DNA of the cancer by analyzing the blood of the patients. So we take 5 mils of blood, that's one standard blood tube, and we can start looking for the cancer DNA that's in the circulation. Then women will have their operation, and we'll take another big bit of tumor as the surgeons are operating. And then they'll have some more chemotherapy, and we'll get some more blood, and we'll get some blood samples as they're being followed up at the end of chemotherapy or when they're having their maintenance treatment. And if unfortunately the cancer recurs, we're going to ask the patients if they would mind giving us another sample that we did for BriTROC1. So in terms of tumour, we'll get a tumour here, we'll get a tumour here, and we may get a tumour there. and we'll get some blood samples so we can monitor what's going on, what we call longitudinally. Next slide, please, Laura. For women who have surgery at the beginning, it's a little bit simpler because, as I'm sure you know, some women start their treatment with chemotherapy, but other women start their treatment with surgery. And so, again, if a woman comes to us with suspected ovarian cancer, we will ask them very nicely, you're going for your operation, would you mind very much if we took a piece of your cancer for research purposes? They'll have their operation, we'll take a bit of sample, and then we will, as I said, when they're having their chemotherapy, we'll take blood samples during the chemo and also during the follow-up. And if unfortunately the cancer relapses, we will be asking for another sample so we can match the sample from before the chemotherapy with one when the cancer comes back. Next slide, please. So what will we do with these samples? So as I've alluded, we will look at the changes in the cancer throughout treatment. How does the cancer get around chemo, and what does it look like when it comes back? Why do some cancers come back and some don't? In BriTROC1, everybody's cancer had come back, so we weren't able to analyze the ones that never came back. Similarly, we weren't able to analyze the cancers of women who were very, very ill and weren't able to give us a biopsy. So we will be able to answer the question here with why do some never come back and some do. We'll be able to answer that question, why are some resistant to treatment and others not? And can we, by analyzing the blood, can we look at really clever ways of analyzing the DNA in the cancer as it responds to treatment that doesn't involve doing biopsies? And the final bit of this is we're also going to try and grow the cancer in the lab. So a lot of what I've described so far is just taking the cancer, extracting the DNA and doing our clever molecular biology. But one thing that we're really excited about is that we've now learned how to grow cancer cells from ascites. As I'm sure many of you know, many women with ovarian cancer develop ascites, fluid in the abdomen that's made by the cancer oozing fluid. And that fluid often contains lots of live cancer cells. And when patients, Laura and my patients, come in and if they've got ascites, we drain it off and 95% of it just gets flushed down the sink. But in fact, it contains a huge amount of incredibly valuable material that we can research on. And for the last few years, we've been perfecting the techniques of how to grow those cancer cells in the laboratory. And we can now do it 90% of the time. So also in this research in BriTROC2, we're going to be taking ascites from women at the time of diagnosis and growing the cancer cells in my laboratory. And so we'll actually be able to say, okay, we'll treat these cancer cells in the lab and we will compare that to treating the patient herself, and we'll compare the two. But those cells will keep growing and we can store them and freeze them and come back to them later on to address more questions. And this is what we call cancer models. So we're going to be able to make new models, new ways of being able to investigate cancer in an ongoing basis because we can grow those cancer cells in the laboratory. And that's really exciting. So next slide, please. How can patients help? So as I hope I've explained, and as Laura's explained, the very best way to investigate cancer, the very best way to do cancer research is to look at the cancers of the patients we're treating. And that's why Laura and I are so enormously privileged, because we both treat patients and we do research. And we're incredibly indebted to our patients who voluntarily say, of course, please, you may have a piece of my cancer. We understand that taking part in research is entirely voluntary. You don't have to do it. And we won't be upset if people say no. They will have absolutely legitimate reasons for not wanting to take part. But for those women who do take part, we are fantastically grateful, because those samples are the bedrock of the research that we do. And all research that I've done, the whole of BriTROC1 would not have been possible without patients kindly agreeing to help us. Ovarian Cancer Action have been very, very generous, both in their time and indeed their funding. And they've created a patient panel who've been helping us and advising us. So whenever we want to do research, we ask patients and say, we've had an idea, what do you think? Do you think this is a sensible idea, or do you think it's a crazy idea? And what could we do differently? And how do patients feel about the research that we're proposing? And that input has been incredibly helpful. And so we're going to do that with BriTROC2, and we're really looking forward to working with Ovarian Cancer Action and with our patients to try and make our research as relevant as we can for the patients we treat. Next slide, please. So the driving force behind my research is I want to improve treatments for the patients I treat now and the patients I will treat in the future. And I want women with ovarian cancer to live longer and live healthier and lives free from side effects and free from disease. And the only way I can do that is through research. And the only way I can do research is through the generosity of funders, such as Ovarian Cancer, and of the patients who generously give their time and their samples, enabling us to take part. So look, thank you very much indeed for joining us. Laura and I will try and answer as many questions as we can in the next few minutes. So if you have any questions, just put them in the chat function or the Q&A function, and somebody somewhere will know how to make that work. And then Laura and I will try and answer them to the best of our ability. So thanks very much.
Thank you so much, Laura and Iain. And that was a fascinating insight into how you're continuing the great work from BriTROC1 by looking at how high-grade serous ovarian cancer evolves and changes over time. And I'm sure the viewers appreciated the molecular biology overview to see how you're looking for patterns within the chaos like you described in the patient samples you use. And also thank you for describing how you include patients in your research as well, as this is really important. And I'm always really pleased when researchers understand the benefit of including patients in their research, so thank you. So we're going to move on to some questions now. So we had a couple in beforehand, and then I'll move over to ones that you've asked during the session. So first off, will the samples be available to other researchers to use outside of Imperial?
Yes, so the BriTROC is a collaborative. So my task, if you like, is to be the curator of the samples. And the whole point of the collaborative is that everything we do is accessible to everybody in the collaborative. So certainly, for example, the cells that we grow from ascites and we store down, they will be available to everybody within the consortium. The genomic analysis, so when we extract the DNA from the tumour and we sequence it, those data are made available to everybody within the collaborative. And when you're doing these sorts of analyses, they're quite technically challenging and quite expensive. And it's always better if one person does, if you like, takes batches of them and does them in relatively large scale. That's much more efficient than giving a few samples to lots of people. So the way we do it is we will centralize some of the analyses, but the data and the results will be available to absolutely everybody to do their work. And similarly, any leftover samples, if somebody says, oh, there's a special test I want to do, we will let them have them. And equally, the cells that we grow, we can freeze down and they will be made freely available to everybody within the collaborative. And so it's an incentive. So we all work together because we work collaboratively, and that's the nature of ovarian cancer research. But we also want to provide an incentive for our colleagues to work with us. And that's why the collaborative has worked well so far, because we share. So we share samples, we share data. Our papers include all the researchers are always included in all our research publications. And so, yes, the short answer is the samples will be made available to anybody within the collaborative.
That's great. And the more analysis of these samples is just going to make any of those findings even more solid and impactful. So that's fantastic. Next question is, how will women included in the trial benefit directly from being involved? And will women from around the clinical centres included in BriTROC benefit from this study being carried out?
It's a very good question. And so some of the benefit from this research, it will be probably quite long term. It's taken us quite a long time. So I hope that we will be able to find new things very rapidly, so that we will be able to understand almost immediately what's happening in that particular patient's cancer. So in BriTROC1, we did identify 10 women who'd had a BRCA1 and 2 mutation that had not been identified prior to them entering the study. So we were able to feed that information back to the treating centres and say, you might not have known this, but we've identified a mutation in this particular patient. We think you ought to go and tell them. And so that was direct benefit. Some of this research is quite long term. And so the benefit may take a little bit of time to emerge. And so I will be completely honest, some of the benefit will be altruistic. So you're helping other women, helping women in the future. But there are some discoveries that we will make rapidly, I hope, that will help women immediately, because it will reveal things about how cancer comes back that will enable us to develop new treatments. So this is quite big picture stuff. I completely acknowledge that. But all treatments that we give women now have arisen from the back of research we did previously, some of it decades ago, some of it very recently. So I hope that women who participate will benefit within a year or two, but it's that sort of timescale. But we hope that there will be information coming out of each individual patient's cancer that will be of direct benefit, that we will feed back to the treating centre, and so that will enable them and their treatment to be changed appropriately.
Great, thank you, Iain. And then we had another question sent in that's quite specific, but they've asked, after extensive chemo, how do you continue the fight if you're not eligible for PARP inhibitors?
So clearly that's a difficult question, and so I'm going to answer this in a general way rather than clearly I don't know everything about every specific cancer. So PARP inhibitors have made a great difference to the way we treat women with ovarian cancer, but there are a group of women whose cancers unfortunately don't benefit from those. So that's what a lot of our research is focused on, and we are now beginning to understand the mechanisms behind those cancers that don't benefit or aren't suitable for a PARP inhibitor. So we know that there's a gene called cyclin E1 that is often amplified in those cancers, and we're now developing specific trials for cancers with cyclin E amplification. We've also understanding that there's another gene in that multicolored wheel that Laura showed earlier on. All the abnormalities on the left-hand side are ones that tend to be associated with cancers that don't benefit from PARP inhibitor therapy. And there's a lot of us focusing on those patients because the PARP inhibitors are not going to be suitable for their cancer, and we have a huge imperative to identify drugs that will. So research in my laboratory from my postdoc, Sarah Spear, is looking specifically at cancers that have a mutation in a gene called P10. Mm-hmm. And Sarah's found that the immune response in cancers that have lost P10 are very, very different from cancers that haven't lost P10. And we will be moving, I hope, towards trials specifically looking at these types of immune populations very rapidly. As I said, there are trials for cancers with cyclin E amplification. We're also looking at cancers that have mutations in a gene called NF1. And again, there are new drugs and new trials coming forward. I will acknowledge a lot of those are not routinely available, but there are trials available in many centres around the country that are specifically looking at abnormalities and mutations in cancers where PARP inhibitors either are not suitable or have stopped working. So again, without wishing to get into specifics of individual cancers, it's important that we investigate clinical trials, especially in those patients for whom PARP inhibitors are not going to be as effective. I hope that answers your question.
Thank you, Iain. And I remember Serous, she gave a fantastic webinar last summer about the PTEN research, and I remember her talking about the macrophages and what's happening there. So that webinar is available on our website if anyone would like to go back and watch that. It was a very good session. And then we have a question for Laura, and how, you're a clinician, how does being involved in research help with your clinical role and treating patients?
So as a clinician primarily, it's really important to have a little bit of knowledge about the research that is happening, because it is helping think about what studies would be good for patients. And also, as a clinician, I'm very involved in all different types of clinical trials. So not just the sample collection studies that we're talking about, but also other clinical trials, and that's all an aspect of research. So yeah, research is a part of, you know, everyday clinical practice really, and will continue to be so.
Yeah.
Great, thank you, Laura. And then we've had 2 questions in, I'm going to put them together. So, will the patient be informed of the research progress, and will the patient know the results of the genetic tests of her cancer?
So the answer to both those questions is yes.
Great.
So, well, yes, and if they want to, respectively. So whenever anybody takes part in research, we ask patients to sign a consent form saying, I've had a discussion with the doctor about this research. The doctor's answered all my questions. I'm happy to take part. And one of the specific things that we mention, and it's in the consent form, is, do you want to know the answers to the genetic tests that we do? In BriTROC2, of the 276 people who consented, 275 said yes to that question. So most people really want to know. So we specifically ask at the start of the trial, do you want to know the results of any genetic or genomic analysis that we do? If the answer is yes, then we will feed back any results to your treating oncologist. Now, when we do our analyses, they're done what's known as anonymously. So we don't know your name. So when anybody takes part in the trial, it's done anonymously. So the treating doctor knows who you are, but when I do my analyses, it's just a number. So I then feed back to, let's say you come from Manchester, I would feed back to Dr. Clamp and the team at the Christie in Manchester and say, patient number 123, who kindly took part in BriTROC, has a mutation in this gene, and they will know who that patient is, and so they will go and inform them if the patient has said yes to that information. So yes, we feed back, and yes, we do updates. And one of the things I'm looking forward to actually is coming back next year and the year after and the year after, and updating you. And I think it's incredibly important as a researcher that we tell people what we're doing. We tell people what's going well, tell people what's going not so well, so that we can keep you up to date. Because I think it's the least we can do if patients have very kindly donated us, allowed us to use their sample. So when you have an operation, even though the sample's in the pathology department, it's still yours. It doesn't belong to anybody else. It belongs to you. And so we would only work on it with your permission. And I think it behoves us, if you've given us permission to use your sample, that we tell you what's going on and what we found. So I will be very happy to come back to this webinar whenever Faye invites me, you posted as to how we're getting on, and give you updates and explain what we found.
Great, and I'm sure the patient representative panel that you mentioned that will be involved in BriTROC2 will be able to advise on things like that and how to keep patients informed in the study, and that's an area that they can really consult and benefit the study. Great, and we've had a really good question that I'm quite interested to know as well. The growing of cancer in the lab sounds fascinating. Is this the future of identifying new treatments, or does response in the lab not indicate response in the real world?
Oh, that's a fantastic question. So there's about, I could answer this one for about a week and a half. I think what we all want, what oncologists like Laura and I want to do, is to meet a patient and say, we will analyze your cancer and we will tell you how well you will respond to our treatment, or even better, which drugs we should treat you with. And that is very, very, very difficult. But growing cancer cells in the laboratory is a very exciting way of moving this forward. So we've now got, as I said, we're have about a 90% success rate in growing cells in the laboratory. And we believe that if you grow them in a particular way, so we grow them what we call in 3 dimensions. So for many years in the lab, we've grown cancer cells flat. And as you may have noticed, human beings aren't flat. Human beings are 3-dimensional. And so we've now learned how to grow cells in 3 dimensions. And how cells respond in 3 dimensions in the lab is much more how they respond in human beings. So the first thing we do is grow the cancer cells on their own in 3 dimensions. And then the next thing we're learning how to do is to add other components in. So if you take a lump of cancer out of a patient, only about half of that lump is cancer cells. The other half is everything else, so what we call the stroma. And that's got blood vessels in it, it's got immune cells, it's got supporting cells called fibroblasts, and a few other bits and pieces. And in the laboratory, we're not only now able to grow the cancer cells in 3 dimensions, we're actually now adding all the other components in as well. So we're really recreating solid cancers in the laboratory. And the behavior of those, you can either call them organoids or spheroids, is much, much more like how patients behave in real life. So the short answer to your question is, do they behave in the same way? Well, partially, and we're getting better and better at correlating or creating things in the laboratory that behave the same as the patient. But coming back to BriTROC2, that's why it's incredibly important that we have a group of women whom we're treating with chemotherapy at the same time as we're growing their cells in the lab, because then I know exactly how the patient is doing at the same time as I'm growing her cells in the laboratory. And therefore we can then make that correlation immediately. So there are many groups in the world trying to do this in different cancers, but the ability to isolate cancer cells from ascites is something we've really now perfected or optimized. And the growing of them in 3 dimensions with other cells, immune cells, stromal cells, fibroblasts, blood vessels, in the lab is something that we're now getting much better at, and that is getting much more predictive or much more representative of what's happening in the patient herself.
Great, and would I be correct in saying that this form of model, using this model, it will take the basic science to patient benefit that much quicker?
Absolutely. As anybody who's ever been in a laboratory, we grow cells. And many of the cells we grow routinely have been growing in the lab for 20 or 30 years. And they're great, but they're not terribly representative of what's happening in the patient. So by growing cells directly from patients and using them in real time, then that will be of immediate benefit, because, as I said, you're investigating something that is straight out the patient, and the patient at the same time is having treatment. You can make those correlations. And this is going to reveal amazing amounts of information for us now and in the future.
Great. I'm conscious of the time. The session is going to come to an end soon, but just one last question. Can the viewers watching today, can they join in the research? Is it available for people to be recruited?
Laura, do you want to answer that one, or shall I?
Go for it, Iain.
So when you set up a trial, there's lots of administrative hurdles you have to get through. So the first thing is you have to go to an ethics committee and persuade them that your research is ethical, and we've done that. Then you have to go to the NHS Health Research Authority and convince them that you've got the logistics to set the trial up, and we got permission to do that last week. Then you have to go to every hospital and say, have you got all the nurses and doctors and samples and patients that you said you did? And we're in the process of doing that. And then you're ready to open. So the short answer is BriTROC2 is due to open in its first centre, I hope, within the next 2 or 3 weeks, which we'll be able, and then we're going to roll this out very rapidly. So as of today, the 15th of March, the study isn't open, but it will be open once we've done all the administrative gumph that we have to do. I very much hope by the beginning of April, and then we will be asking as many women as we can to help us with the research. So I hope within a few weeks.
Great, and we're getting comments through just saying thank you for an excellent webinar and an excellent session. So thank you so much to the both of you, and I think we'll leave it there today. And thanks again to everyone for joining us this evening. And if you'd like to see what other research sessions we have coming up, please visit our website. And I'd be really grateful if you could complete a quick post-webinar survey with your thoughts on the session via the email I send out after. And this helps us to make the sessions more relevant and engaging for you all. So thanks, massive thank you to Laura and Iain. That was a really interesting session, and I hope everyone has a great evening, and hopefully see you at our next research webinar. Thank you, everyone.
Thank you very much indeed.
Thank you.